Welcome back. In the last session, we were discussing the enzyme aromatase, whose function is to convert the androgens into estrogens. Okay. And if you can stop the biosynthesis of estrogen, then you can have an anti-cancer drug. That biology is now has been discussed. Now, the question is how the androgens are converted into estrogen. The reaction is nothing but removal of a methyl that you have to do in order to aromatize the A ring. Okay. First, the methyl is oxidized to CH2OH. Who does the oxidation? It's an iron mediated, and oxygen is the co-substrate, and electrons come from the NADPH. Okay, NADPH is electron donor, oxygen is a co-substrate, and then iron is the mediator of electron transport. Okay. So, first it goes to alcohol, then another round of oxidation, it goes to the aldehyde, and from aldehyde we will start. Okay. The aldehyde, where is the aldehyde? This is the aldehyde. Okay. This is the aldehyde, and this is what is the aromatase enzyme. It has got this porphyrin 4 nitrogen, typical 4 nitrogen system, which is linked to the ferrous. So, the iron is in the ferrous state. Okay. And then the aromatase have a cysteine residue, and the iron is coordinated to the sulfur. Okay. You know that when the iron is the ferrous, it is a pyramidal, it is a pyramidal structure, but when it becomes the Fe3, it becomes square planar. Now, how does it become sp3? By donation of, by forming a bond with the oxygen. Oxygen is triplet. So, now iron gives one electron to the oxygen, and this oxygen donates the other electron to form this iron oxygen bond, and this is O dot. Now, the NADH transports one electron. So, it becomes O minus, and this O minus attracts the aldehyde carbonyl. So, what will happen? This becomes, this is the aromatase. I can write it here, means you do not have to write that big. This is still in the ferric state that is now attached to the oxygen, then another oxygen, then OH, and you have this part, sorry, not this, just a second. So, you have, this is the other A ring and this is the B ring, A ring, B ring. Now, you have double bond O here, and you have one hydrogen, here. So, this is the situation, and the C ring is attached here. Okay. This is the fate of the methyl. Now, you can write this as OH, sorry, there was a double bond to start with here, there was a double bond. So, you can form this enol now. Okay. As you form the enol, this hydrogen becomes acidic, and that comes here, and this bond goes out, that comes here, that actually basically takes the hydrogen. So, this takes the hydrogen, that comes here, that goes there. So, this is basically what? This is nothing but formate, and this is the iron, this ultimately this will be iron OH, iron 3 OH, that comes out, and in the process this becomes what? A double bond that is now incorporated to make it an aromatic ring. So, this is the mechanism of your aromatase. So, if you want to utilize, make inhibitors of this, now there are these drugs which are available in the market. There are non-steroidal drugs also. These are not no linkage with the steroid or no relation with the steroid, as if apparently, but they are inhibiting the aromatase. Okay. And these are kind of this steroid, these are steroid type molecules. How they act as inhibitors? I think that I will show you in the next slide. Basically, in all these molecules, you have this another extra double bond. Here it is a methylene, here it is a extra double bond, here this is already present in the in your androgen. Okay. like this androgen was already a double bond. So, what happens if you put double bonds, now that creates potential sites for attack by the nucleophile, any nucleophilic amino acid in the enzyme. Enzyme is the aromatase enzyme. So, this is the aromatase enzyme. This was doing the oxidation of the aldehyde, the ferric oxygen. Remember the last step of this ferryl-oxo species, not the ferryl-oxo, the breakage of this peroxo linkage. So, that was the intermediate here, and then that breaks. So, basically that what is happening here, but in order to do so you do not have to lose the hydrogen. The hydrogen is not there, of course, this is a vinyl hydrogen. So, you cannot abstract the hydrogen. The nucleophile in the form of an enzyme residue, enzyme amino acid residue, that attacks here, this goes out here and that breaks. The format comes out, iron comes out as the same thing, the Fe3 which, but the aromatase enzyme is now hooked here. So, this is nothing but a suicide inhibition, a case of suicide inhibition. Okay. Similarly, I think this is clear. Similarly, for the other compounds which look that they have no relation with the steroid, what they do, they actually chelate to the iron and stop the oxygen to bind to the iron. Okay. So, they actually block the oxygen to bind to the iron. Oxygen, remember, is the co-substrate. So, thereby stopping the aromatase enzyme from working. The end of the anti-cancer drug development that we have discussed so far. So, basically to summarize what we have done, first we discussed what are the what is basically cancer, that you have a problem with the cell cycle functioning. Okay. There is no programmed cell death, which was called apoptosis. So, apoptosis phenomena is not working in cancer, and for preparing the cell for to different stages. So, there are proteins which are called cyclins, and there are kinase enzymes which are called CDK, and they actually get the signal from outside of the cell, that and then collect the signals and then see whether it needs to be grown, it needs to carry forward, or there is no need for cell growth for the time being. So, once it receives all those signals, it pushes it activates the growth factor and then pushes the cell forward in the forward direction. Forward direction means from G1 to S, S to G2 and G2 to M. M means the mitotic phase. Okay. And if there is no requirement of cell cycle, it goes to the resting stage, that is called G0 state. And if there is some problem with the DNA that is made, then the cells are destined to die. So, that is what is the apoptosis. Okay. When the enzymes cannot repair whatever the problem with the DNA, then it will ultimately throw that cell into death. Okay. In cancer, that programmed cell death does not happen. So, one good target is the cycling dependent systems. Okay, I told you. So, there are many kinase enzymes inhibitors which are now available in the market as anti-cancer drug, although we have not discussed that. But later on, then we stated what are the what could be the targets for cancer, and we found that the DNA could be a very good target, because that is the commander in chief of the cell cycle. So, you can use alkylating agents, you can use chain cutters, you can use intercalators, and we have given an example of all these agents. Then we went to discuss the HDAC, that means the histone proteins, how can you modify them and then or perturb them not to work efficiently, and HDAC inhibitors as simple as SAHA, that is a hydroxamic acid which chelates to zinc, and HDAC is, by the way, a zinc dependent enzyme, hydrolytic enzyme. So, it stops the HDAC from working. So, that was one new development, and then we talked about the aromatase inhibition. Aromatase, that estrogen production from androgen, and that can be stopped. There are many drugs that are available in the market today. So, we finish with that the anti-cancer drugs, and now we will move to some other topic, which is the another very important agents that are taken by which are sold to a large extent. They are called anti-ulcer drugs. We suffer from acidity quite often, and today there are very good drugs. Earlier days, when people have hyperacidity, they used to suffer from what is called peptic ulcer, ulceration of the GI tract. Okay. And but now today we have very good drugs to combat the formation of ulcer, because we can take care of the acid. Earlier days, what was the treatment for acid is to use bases. Okay. Very simple, that if acid is formed, then you have to add base to neutralize it. Okay. And that is the genesis of development of Gelusil or whatever. So, very similar type of agents. These are hydrated alumina magnesium oxide that neutralizes, but that does not stop the production of HCl in the stomach. Okay. Because simple Le Chatelier principle says that the more you neutralize the HCl, the more HCl will be formed in the body. Okay. So, we should really know what actually is causing this acidity. If we know what is the causative, what is the reason for having hyperacidity, then we can develop in a much better antacids, which are basically targeting the actual reason for hyperacidity. Okay. So, let us start that. So, in order to do that, what we need to know, first of all, as I said, acidity. Right? What is happening? Let us see. So, in order to study this aspect of medicinal chemistry, first, cause of acidity. Number 2 is then target identification, okay? Because these are actually intimately connected, cause of acidity and target identification, and then develop drugs. Now, acidity, before we proceed, acidity also can be caused by some infecting agent, which is called H. pylori, is one microorganism which also can cause hyperacidity, and the person who has developed that H. pylori is the causative agent of ulcer, he got a Nobel Prize. He is an Australian, and he himself took that H. pylori tablets into his body, and then trying to see that whether it develops ulcer or not. Okay. So, that is the, in the quest of discovery, they can even risk their own health. Now, we are not talking about H. pylori, that is actually given here, no? H. pylori. H. pylori, now we have antibiotics, antibacterial agents. So, once it is known that H. pylori is the causative agent, then you can tackle that, okay, by utilizing amoxicillin and then an antacid, what we are going to discuss, and also it is a cocktail of agents that is given to tackle the H. pylori. But we are talking about something which is what is the actual reason or having acidity. Remember, acidity is required to maintain a particular pH, because some of the enzymes like pepsin, which works at an acidic pH. So, if you want to digest food, you cannot have that acid will not be produced. That is not also good, because you need acidity, a perfect pH, so that the enzymes work and breaks down the peptide bonds, and one example is pepsin. So, acidity will be HCl production is needed, but at a controlled rate, not at a high rate. Okay. Now, what how so let us first discuss what causes acidity. Now, this is the stomach, a rough cartoon of the stomach. Okay. In the stomach, in the outer lining, actually this is shown here, in the outer lining there are some cells which are called parietal cells. In the outer lining. Okay. These parietal cells, if you expand it, they look like this. This is what is the, see, this is circle, in the circle you say parietal cells. The parietal cells basically is a connection between the outside of the stomach, whatever is flowing here, and inside, because what happens, it can absorb something and it can, there is a gap here. So, through the gap it can push the ions or other small molecules, whatever is required, it can do that. Achha. One thing that it does is that it has got what is called a proton pump. Means proton pump means it pumps in protons. So, basically you have the stomach, there is a parietal cell which has an opening here. Okay. A parietal cell like this, it has got an opening. So, its job is to pump proton inside the stomach. Okay. Where from the chloride ions come? So, that is the cause of protons, but where from the protons come, that is one. Where from the chloride comes? Actually, before that there is one stage, that stage is carbonic, there is an enzyme called carbonic anhydrase. Carbonic anhydrase is basically water plus carbon dioxide becomes carbonic acid, which is a weak acid, but it dissociates into H plus and bicarbonate. Okay. That is the source of H plus, and bicarbonate is replaced by, bicarbonate does not enter the parietal cells, the proton enters along with chloride, and to maintain balance bicarbonate is pumped out of the system. Okay. So, basically what ultimately remains is H and Cl. So, that makes HCl. Okay. Now, what signals that this proton pump has to work? There must be some control mechanism by which this proton pump works. Now, again I repeat, this proton pump is basically, why these are called pumps? Because pumps are what? It releases air on one side and then brings water. Okay. That is one type of pump. Proton pump means it puts proton inside, but you cannot have it has to be, it has to taken out some counter, the ion also, this is positively charged, your stomach cannot be positively charged. So, you have to take out some positive ions, and in fact, it takes out the potassium ion out of the stomach. Okay. So, that is why this is called H plus K plus proton pump. H plus K plus proton pump. The first cation means it is going inside, and the second cation means it is going outside. Okay. So, this proton pump, where from the energy comes? This is an enzyme which hydrolyzes ATP. Energy comes from hydrolysis of ATP. So, it is a proton pump which is nothing but a hydrogen H plus K plus ATPase. Okay. So, that is the proton pump. What regulates the proton pump? Three things. One is histamine. What is histamine? Histamine is the decarboxylated version of histidine. If you decarboxylate histidine, you get histamine. Okay. Like dopamine, we have read dopamine, that is the decarboxylated version of L-dopa. Okay. Similarly, histamine. So, histamine has a receptor on the parietal cell. There is another neurotransmitter. Histamine, by the way, you can call it as a hormone, because it works at several places. Another neurotransmitter, acetylcholine, we have already read that. So, there is a receptor for acetylcholine, and there is a receptor for a peptide which is called gastrin. And so, any of these, if they bind to this receptor, that sends a signal to the proton pump to work and then push the H plus inside the stomach, and that causes acidity. So, now you know what causes acidity. So, what will be your strategy? Either to stop the acetylcholine from binding here, or to stop the gastrin from binding there, or to stop histamine from binding here. Three strategies. I think this strategy was not successful, it is a big peptide. Acetylcholine, people started first looking at it, that inhibitors of acetylcholine, you know, there are inhibitors of acetylcholine, or your antagonist you can use. There is a third receptor H3, which is present in the CNS, central nervous system. If it binds to H1 receptor, it causes allergy. If it binds to H2 receptor, it causes acidity. If it binds to CNS, it causes sleepiness, drowsiness. And I think now you can equate that why when you take anti-allergic tablets, you also become sleepy, because your drug is not very specific. It is an antagonist to H1. It is also reacting with the H3 receptor. It is binding to the H3. So, if it binds to the H3, then the problem is that you get drowsiness. Okay. Now, today we are talking about this H2 receptor, the acidity. Okay. So, let us discuss it a little bit more detail, slightly more detail. So, we now talk about, we forget about the acetylcholine receptor or gastrin receptor. So, this is our stomach part. Okay. This is the parietal cells. Okay. Now, in the parietal cell you have this receptor, which is the H2 receptor, histamine H2 receptor. By the way, the receptors are named according to the hormone or neurotransmitter, like adrenergic receptor, which is the receptor for adrenaline. This is histaminergic receptor, histamine. Okay. Cholinergic receptor, choline receptors. So, this is histaminergic receptors are three types, H1, H2, H3. We are talking about H2, which causes acidity. Now, if the histamine binds here, there is a reaction that takes place at the parietal cell, that is ATP converted to cyclic AMP. You have seen this earlier also. Cyclic AMP is called actually that was cyclic GMP earlier. In the G protein coupled receptors, there was a formation of cyclic GMP, which was a second messenger, I told you, because that goes from here and then takes the message to somewhere else. So, this is the primary messenger, histamine, and this is the secondary messenger, cyclic AMP. So, ATP, cyclic AMP is nothing but this O phosphate, and it is a 5 prime position, that O. This is cyclic AMP. So, this is A. Okay. So, cyclic AMP is formed, it goes to the pump now and says that now you have to start working. So, it now pushes the H plus here and potassium plus inside. Okay. So, that is the thing. So, now how to stop this acidity? You can stop this, you can target the histamine receptor, that is one, or you can also look at this pump. You can stop the pump also, because that is also an enzyme. Okay. There are very good H2 antagonists. You know the names, ranitidine, or the first one that was discovered was cimetidine. Now, you have ranitidine, you have famotidine, you have, I think this is now, this has gone out, the first generation. Now, ranitidine is still very successful drug to develop for stopping the acidity, controlling the acidity. Okay. So, that works by as an antagonist of histamine H2 receptor. Okay. Very simple. Now, let us talk about this one, the pump. The pump is stopped by a drug which is also very well known, omeprazole. Now, the anti-ulcer compounds are basically omeprazole, rabeprazole, lisinoprazole, then pantoprazole, all these compounds. They are actually inhibitors of this proton pump. What is the mechanism of that? the mechanism of proton pump is, so this is actually this it started from H1 receptor. This was the H1 receptor. How this ranitidine was discovered? First they started looking at the receptor active sites. Okay, what are the locations where it binds? Okay. So, initially they developed H1 receptor. All these anti-allergic drugs were first discovered, which are H1 receptor antagonists, and now we have this is pheniramine, this is diphenhydramine, and this is a much better one which is called cetirizine, and then you have this levocetirizine, the L isomer is what is called levocetirizine. Okay. But we are not talking about, but they actually bind to the H1 receptor. Levocetirizine is even specific, which does not interfere with the H3 receptor. That is why this is so successful. It causes less drowsiness. I think I told you about the source of HCl. This is the source of, this is also I told you what happens in the parietal cells. Now, two strategies for antacid. One is for managing the hyperacidity, H2 antagonist, another is proton pump inhibitor. H2 antagonist, how was it discovered? See, when they initially nothing was known, but they thought that okay, there could be some receptor. Why this structure, histamine structure, if you look, it will be present as NH3 plus here and this imidazole. Okay. So, there can be some interactions here and then some electrostatic interactions here. So, possibly there is a CO2 minus here, and there could be some hydrogen bonding effect, which forms the hydrogen bond to nitrogen. But the problem is how to really develop selective agent, that not an anti-allergic, but only goes to the H2 receptor. Okay. So, by that we are just making the discussion short. What happens if you try to utilize this type of system, only this type of this amine and an imidazole, you are not satisfying what is called the Lipinski's rule. Remember, Lipinski's rule is basically the initial screening of a drug like molecule. 5 hydrogen bond donors, 10 hydrogen bond acceptor, that is Lipinski's rule of 5. So, they are also important. Pharmaceutical companies, they will work on Lipinski's rule. We academicians, we think that Lipinski's rule is not correct. Yes, it is not the ultimate, but it is a way of screening out structures. Okay. So, based on these, what was made or what was found, that if you attach to the histamine another binding agent, that means it was, I think it is just by extrapolation, they thought that there may be other binding sites in the H2 receptor. So, what they did, that in addition to the histamine type molecule, they attach something else which can also form a hydrogen bond at the other binding site, although they did not have some idea initially, they do not have any idea that where is this binding site, but they have this hypothesis that there are basically 3 sites in the H2 and 2 sites in H1. So, if you want to exploit the 3 sites, what you have to do? If you take only histamine, they will go to these 2 sites. The other third site is remain vacant. Okay. If you now attach something which can interact with the third site, then you will get very specific H2 receptor binding molecule. Okay. So, ultimately they came out with these type of molecules, cimetidine I told you, then ranitidine, this is the structure, and famotidine, this is the structure. Now, the question is where is basically they are binding. First they actually try to satisfy the Lipinski's rule, they put the sulfur inside. Okay. So, another heteroatom they put, lot of nitrogens you can see now. Okay. So, they put lot of nitrogen donors. Okay. And then they have acceptors also, this is donor, this is acceptor, this is donor. Okay. This is acceptor, this is acceptor, this is also acceptor. So, this is what is the, I think that I told you. Now, the structure, the crystal structure, possibly the structure shows that or maybe this is by modeling studies that they found or it is hypothesized that an aspartate is here, which forms a hydrogen bond interactions here, a threonine in the active site that forms hydrogen bond with the remember this is what is the structure of ranitidine. Now, this is the cimetidine, actually that structure is for cimetidine, a C double triple bond N, and let us see C triple bond N goes to the threonine, this goes to the aspartate, and that goes to another aspartate. I told you there are 3 binding pockets. So, one binding pocket takes care of the imidazole, that you cannot change, and this is the threonine and that is the aspartate. Okay. Okay, now let us come to the final part of this anti-ulcer drugs, that is the discovery of omeprazole. Remember, omeprazole is the one, these type of molecules they are called proton pump inhibitors. Okay. There is a series of drugs, omeprazole, rabeprazole, pantoprazole. We just talk about the omeprazole. This is the molecule called omeprazole. Okay. It has got a sulfoxide, it has got this benzimidazole and a pyridine nucleus. Now, what happens here? It exploits the hyperacidity of the stomach. So, first it is the basic nitrogen of the imidazole that gets protonated. So, once it becomes protonated, this nitrogen is nucleophilic, nitrogen from the pyridine that attacks here and this comes here. So, basically what happens? You get a molecule like this. Now, this is a sulfoxide. So, that can also abstract a hydrogen and which results in the breakage of the carbon sulfur bond. This is nothing but what is called a Pomeroy rearrangement. Pomeroy rearrangement is basically a nucleophilic ring opening or a hydrogen loss via hydrogen loss, ring opening of the carbon sulfur or breakage of the carbon sulfur bond. Okay. So, once that is broken, it will be say SOH, this is NH, this is double bond and that nitrogen is plus. Now, this again comes here, that goes and removes the OH. So, you get a cyclic compound like this, but this is now the sulfur. Look at this sulfur, this carbon nitrogen sulfur bond is very vulnerable, because this nitrogen can be protonated again and that makes this sulfur a site of nucleophilic attack. So, the enzyme means the proton pump. The proton pump with a active serine SH, cysteine SH that attacks this sulfur, this comes here and that goes out, that takes the proton. So, basically what happens here? You get a stable enzyme and transformed substrate. It is a covalent bond. So, this is a case of suicide inhibition. Okay. So, basically now to conclude this anti-ulcer chemistry, that there are basically 2 strategies. One could be on the H2 antagonist, another could be the proton pump. H2 antagonist, lot of structure activity relationship was done, and then they realized that there are 3 binding pockets and they have utilized these 3 binding pockets in order to have very specific H2 antagonist, which does not bind on H1 or H3. Okay. So, that is the genesis of development of ranitidine, cimetidine, those type of drugs. And then the proton pump inhibitors, they are basically these proton pump inhibitors are suicide inhibitors, and it is catalyzed in presence of high acidity, because it is catalyzed by the first step is basically protonation, and that protonation is the driving force for formation of a enzyme transformed substrate via a covalent bond. So, that is called suicide inhibition. So, I think that is the end of the anti-ulcer chemistry developments, and there are a lot of now today peptic ulcer is almost gone. People are not suffering from peptic ulcer, which was not there in the 30 years back. Lot of people were suffering from peptic ulcer, but with the advent of omeprazole, ranitidine, now this ulcer problem is almost gone these days. Thank you.